Hi, hello everybody. This is Dr. Srivani Sesham. Welcome back to my channel. If you have not yet subscribed to my channel, please like, share and subscribe. Today I am going to present to you another important topic in genetics, epistasis. So let's now first define epistasis. Epistasis is the interaction of non-allelic genes in which one gene masks, inhibits or suppresses the expression of the other gene. The gene that suppresses the other gene is known as inhibiting or epistatic factor, and the one which is prevented from exhibiting itself is known as hypostatic factor. Although it is similar to dominance and recessiveness, in epistasis the two factors occupy two different loci. Dominance involves intragenic or interallelic gene suppression, while epistasis involves intergenic suppression and is of two types. One is dominant epistasis and the second one is recessive epistasis. Now let us see each one separately. Dominant epistasis. In dominant epistasis, out of the two pairs of genes, the dominant allele of one gene masks the activity of other allelic pair, dominant or recessive, at other loci. Like if the gene A is the epistatic factor, then it suppresses the expression of gene B which is present on a different loci, whether it is in the dominant state or in the recessive state. Here you can see clearly in the representation, beside recessive I have written capital as well as small letters, that is recessive. In that character can be a dominant one or a recessive one. Then since the dominant epistatic gene A exerts its epistatic influence by suppressing the expression of gene B, that is capital B, in the dominant state or small b which is in the recessive state, it is known as dominant epistasis. Now let us take one example of dominant epistasis in case of dogs. So in dogs, white coat color appears to be dominant. It develops due to the action of epistatic gene capital I, which prevents the formation of pigment controlled by the hypostatic gene capital B. The hypostatic gene B produces black coat color, while its hypostatic allele small b produces brown coat color only when gene I is recessive. So if they have to express the color, the allelic pair which is present should be in the recessive state, then only they can express their color, otherwise they are the hypostatic factors. The epistatic factor masks their expression, so they cannot express their body color. So the progeny of dominant gene I does not allow them to function and it results in white coat color. When two white coat dogs are crossed, they produce white, black and brown coats in the ratio of 12:3:1. The white dogs in this case possess genes for black or brown color but does not produce the pigment because of the presence of gene I in dominant state. Here gene I which is in dominant state is the epistatic factor which covers or masks the other gene from expressing the color. Okay, so the depiction you can see here, the first parental generation, brown dog is crossed with a white dog. The brown dog's genotype is capital B, capital I, and white dog is small b, small I and small I. So the gametes are formed which consist of capital B and capital I and small b and small I. So whenever, as I have told you, since it is dominant epistasis, if capital I is present, the coloration cannot be expressed. So the progeny that is formed in the F1 generation are all white in color, that is the dogs have white coat color. Okay, so when F1 is selfed, that is F1 is crossed with the F1 progeny again, then the gametes formed are capital B, capital I, small capital B, small b, capital I, small I from both the parents. When two white coats are crossed, dogs are crossed, they produce white, black and brown coats in the ratio of 12:3:1. So here we can see the depiction of dominant epistasis in case of dogs, coat color being depicted in the Punnett square. So the male and the female gametes are shown in two different rows, where a crossing between different gametes when it takes place, resulting in the formation of progeny either showing white coat color or black or brown. But if black or brown are to be exhibited or expressed, the dominant gene capital I should not be present, because capital I which is present, which acts as an epistatic factor which suppresses the expression of the other gene. So wherever we find the presence of capital I in the complete crossing overs which are shown here, the resulting outcome is white coat color only. So whenever there is absence of capital I and presence of small I, we can see the coat color being expressed, because the suppression activity of the capital I is not present. The epistatic factor is absent. So capital B, small I. If you notice here, whenever capital I is absent, black color is expressed. Or capital B, small I. Since capital B is a dominant one, so black coat color is expressed. Like that, you have three black coat colors being expressed and one brown which carries both the recessive alleles. Since small b expresses brown color with the absence of the capital I, which is the epistatic factor, only small I is present, so brown color is expressed. So the dominant epistasis ratio is 12 whites is to 3 black is to 1 brown color. Now next one is the recessive epistasis. This epistasis is due to recessive gene, so it is termed as recessive epistasis. That is, out of the two pairs of the genes, the recessive epistatic gene masks the activity of the dominant gene of the other gene locus. So the dominant gene A expresses itself only when the epistatic locus capital C also has the dominant gene. Otherwise, if the epistatic locus has the recessive gene small c, then gene A fails to express. Hope you are able to understand. Here the recessive gene small c is the epistatic factor. The recessive gene is acting as the epistatic factor, suppressing the expression of the dominant gene. That is why this is termed as recessive epistasis. Now let us see an example for recessive epistasis. Example is the coat color in mice. In mice, agouti color characterized by banding of heads is controlled by gene A, which is hypostatic to recessive allele small c. The dominant epistatic gene capital C, in absence of A, gives black colored mice, and in presence of dominant gene capital A gives agouti, but dominant gene A is unable to produce agouti color in the absence of gene small c. Recessive gene C acts as epistatic gene here. So we can see in the depiction of the recessive epistasis here in the Punnett square, where three different color codes are formed in mice: agouti 9, black 3 and albino 4. So wherever you can see the presence of the recessive gene small c is suppressing the expression of the capital A, the coat color is not expressed. So we can see here formation of 4 white or albino, that is no pigmentation is present, albinos are formed. Rest of the places wherever the recessive gene is not present to suppress the expression of the dominant genes, there all we can see the coat color being expressed. Thank you for watching my video. Please like, share and subscribe. If you have not yet subscribed, Please do subscribe and hit the bell button so that you can get the fresh notifications whenever subject videos are posted